Okay, good morning. I hope all of you had a nice long weekend. So as a reminder, we have our exam coming up this week on Wednesday. So let's go ahead and take a look at our calendar and see what's going on here. Hopefully this didn't sneak up on you because it is kind of strange, right? We came back from spring break, we had a short week. And then because this is also a short week, since Friday is Good Friday, that kind of makes our schedule a little condensed. And so we have to have our exam on Wednesday instead of on Friday. So here we are we're going to talk about today sources of DNA damage and talk about some mechanisms for resisting cancer. Wednesday will be our second exam that will cover chapters 7 through 22. Not 22, 7 through 12. Sorry, I was looking somewhere else on the on the calendar. So unfortunately, we will not have the opportunity to actually dedicate a class session to reviewing, but I do have review slides that I can share with you, and I may still have the recording of my review lecture from last spring, which I can find and maybe put on YouTube, and maybe that'll be useful as well. So I'll take a look at that. You do have a quiz due this week also. My feeling is that oftentimes it's good for you guys to take a quiz as part of your preparation for the exam, since it helps you sort of identify certain concepts that are important as you're studying. So I will make the quiz available right after our class today, because I wanted to make sure we get through this chapter 12 material. And then I'll have the quiz due Wednesday as normal at class time. So you just get through that as part of your studying. We take our exam on Wednesday, and then we don't have class on Friday. As a result, I've decided not to also pile on an additional homework assignment, because originally this week there was also supposed to be a homework assignment to help you study. But but since the exam is right in the middle of the week, I kind of figured, yeah, that's a lot of work. All right, so it's crossed out here. So I will post an updated calendar that has this stuff all correct on it. All right, so don't worry about the homework this week, but do worry about taking the quiz by Wednesday and being prepared to take the exam on Wednesday, chapter 7 through 12. All right. Let's continue where we left off last time. We were talking about how there are different sources of mutagenic threats that our genome inside of our cells are always subjected to every single day. And I said that one way of thinking about where these threats come from is by considering that many of these things happen just as a function of the cell being alive and doing different types of processes. Last time we looked at the first example here, mistakes during DNA replication that take place during S phase. And I think that's something that is probably the least obvious or the least intuitive to think about. But every time your cell, or a cell, let's say a stem cell, undergoes mitosis, leading up to mitosis, of course, it has to replicate its entire genome, all 3.2 billion base pairs, and it's got to do it as accurately as possible, right? Because we said last time that any mistakes that are made in the replication, boy, from that moment on, when that stem cell, right, divides and you get two daughter cells, right, it's quite possible that those mistakes now permanently become baggage, right, that are part of the stem cell. And for the next several, potential decades of your life, that stem cell is always going to have that mistake, and it's going to have different other mistakes piled on top of it. Right? So the replication process of DNA is really impressively efficient, right? Starting with the efficiency of the DNA polymerase holoenzyme that's actually copying the template DNA. Remember, the mistake error rate of that is really, really low, right? And so, and also it can proofread it, it can move backwards. And if it makes a mistake and misses something, then you have other mismatch repair enzymes that come. And those things together makes the error rate about 1 base per billion bases copied, right, end up being wrong. And so that means in your entire genome of 3.2 billion bases, you get on average about 3 mistakes that are made and passed on just from the DNA replication process itself. Right? Let's take a look at these other factors here. Number 2, endogenous biochemical processes. In other words, in addition to the DNA replication happening involving, proteins, enzymes, things like that, of course, your cell has all these metabolic processes happening as you're breaking down molecules, releasing energy, assembling molecules, etc. And there's going to be lots of intermediate products that are made. Many intermediate products are things that end up being discarded because they're just sort of like a little byproduct of the molecular pathway trying to make something else, right? And those intermediate products, those little scraps and things of your metabolic process, many of those are highly reactive, right? Because they represent like a piece of a molecule that was broken off and it might want to react with something else. And then thirdly, today we'll wrap up talking about exogenous mutagenic factors. And I said last time, I think it's usually the exogenous mutagenic factors, the outside factors, exposures that we usually think of when you think about, things that cause mutations, right? So we'll get to that as well. All right, so let's start today talking about endogenous biochemical processes. Things fall apart, right? Even though our molecules are what we call metastable, meaning life evolved to use certain biological molecules and treat them importantly because they are stable enough that they won't just spontaneously all the time break down, because life, that would not work very well for life if the things that we depended upon, like proteins, carbohydrates, lipids, just fell apart on a moment's notice just because they felt like it, right? They will stay together. Right? But of course, we're still just talking about atoms bound together with other atoms. So there are times when, especially if you're talking about something huge like a chromosome, right, a big string of potentially millions of nucleotides, things are going to just kind of fall apart occasionally. Right? So a common thing that happens is loss of purines. Here is a strand of DNA. All of you are familiar. This is a nucleotide. So this is a guanine. So the nitrogenous base, deoxyribose sugar here, and a phosphate group, right? So this is the phosphate backbone of the DNA, and you have this guanine sticking out. Now the guanine, it's a pretty bulky, purines in general, pretty bulky sort of groups, right? And so it is possible that you can actually have water molecule come in here and break off the guanine, and the guanine floats away, right? And you end up with a depurinated deoxyribose. Now, hopefully, your cell can detect that there's something wrong here. And hopefully, right, based upon the fact that across the way from this DNA strand is the other DNA strand, because it's the double helix, and hopefully on the other side, right, you'd have the C, right, the cytosine, and that you'd have enzymes that would be able to recognize, there's a cytosine on the other side, so this is probably supposed to be a guanine here, and hopefully it can fix it, right? But if it doesn't, then you've actually lost one of these bases now in your genetic sequence. Right? How often does this happen? Right? Again, right, if you look at the atomic structure, you realize, it doesn't look like it's a very stable molecule, right? So it's estimated that you lose about 10,000 purine bases per cell every day in your body, right? And if you're talking about trillions of cells in your body, each one of which losing on average 10,000 purines just in the course of you being alive another day, right? Hopefully most of those get patched up and repaired, but some of them might not be. Right? What about instead of the base falling off, what if the base gets modified in such a way? Right? So here we have examples where the amine groups that are part, or the amino groups that are part of bases sometimes can get removed. So for example, if you have a cytosine here, so this is the base part, right? This is not showing the ribose or deoxyribose sugar. It's not showing the phosphate. We're just focused on the nitrogenous base. So cytosines can have the amine group up here removed, right? And you end up with just an oxygen there. And what does it become? The cytosine has actually become a uracil. Right? You can have an adenine over here, right? Again, another purine, right? And so you can actually have the amine group removed, and so it's replaced by an oxygen up here. This becomes hypoxanthine. Right? Guanines, you can have the amine group removed over here, right, replaced by an oxygen, and become a xanthine. This one is really interesting. This is 5-methylcytosine. We've talked about 5-methylcytosine before in the context of DNA methylation. Because remember, when you have a CpG site, right, so a cytosine followed by guanine, and they're held together by a phosphodiester bond because it's meant to show you it's on the same strand of the DNA, those cytosines can be methylated. Right? So here, that's what this methyl group is. It's been added to the cytosine. Right? Compare that to the plain old regular cytosine up here, right, which is supposed to have just a hydrogen over here. So this is a methylated cytosine, 5-methylcytosine. And remember, in places like CpG islands, in promoters of genes, these CpGs might become hypermethylated, and as a result, that causes the histones, the nucleosomes, to hang on to the DNA tighter, and the DNA, the chromatin, compacts. Right? So we said that hypermethylation of CpG islands, right, chains of CpGs in the promoters, is an effective way for our cells to more or less indefinitely keep a gene shut down. Right? So as part of differentiation, when we have cells that start off as stem cells, and as the stem cell divides to repopulate things like differentiated cells, certain genes in the genome that this particular cell type has no business worrying about expressing, right? Many of those genes are more or less indefinitely shut down through CpG island methylation at promoters. Right? We also said that during cancer, the progression of cancer, you can actually have the reverse of that, where you have certain genes that should be shut down due to DNA methylation, that improperly now the methyl groups get released, and suddenly those cells start expressing certain genes, again, that may contribute oncogenically towards things like loss of proliferation, for example. But to get back to this story here, look what happens if you have a 5-methylcytosine, right? And then you make a modification over here. Right? You remove the amine group, the amino group, right? Replace it with oxygen. This becomes thymine. Right? This becomes just plain old ordinary thymine, which obviously your cells tolerate very well, right? Because this is just a T. So that's why evolutionarily, over evolutionary history, eons and eons, random CpG sites in our genome have been converted from cytosines to thymines, right? Through many, many millions of years of just this process of deamination. And that's why if you randomly pick a spot in your genetic sequence, right, you would find fewer CpGs than by chance. But all the CpGs that are by promoters, those are conserved in evolution. So there are mechanisms to protect that those CpGs stay as CpGs. So what this lets you think about then is how the identity of certain bases can just be changed just by this process of deamination. And as part of metabolic processes, I mentioned that there's going to be lots of intermediate molecules that are just kind of floating around as bits and pieces of sort of waste products of different types of molecular interactions, right? Many of these intermediate molecules are highly reactive because they contain oxygen groups on them that would very willingly and happily interact with other molecules and maybe try to oxidize those other molecules, thereby changing their proper shape. So here's an example. So here's a base, right? So this is deoxyguanosine. Right? It can be oxidized. Right? So in this case, you have some sort of a molecule that had a available oxygen on it, floated by, right? And it underwent this reaction, and it changed this into a 8-oxodeoxyguanosine, right, which is now no longer the proper base that should be there. It's supposed to be a G. So the problem here is that base in this 8-oxo-DG form can improperly form hydrogen bonds across with adenines. All right, so if you had this red molecule on one strand of DNA on one side of the helix, and then you had this brown base as part of the other strand of DNA, you can see that you might end up with improper base pairing, right? Not your normal traditional Watson-Crick base pairing. You'd end up with this strange sort of mispairing that would cause a defect in the shape of the DNA. And this was due to reactive oxidation species attacking or damaging this guanosine. Right. So these reactive oxidation species, just in the process of the cell breaking down large molecules as part of our catabolism of large molecules, you're going to have all these little random intermediate pieces. And you better believe a lot of them have these oxygen groups on them, right? That could then potentially damage other molecules if they bump into them. So we must have evolved ways of trying to neutralize these reactive oxidation species to prevent them from building up inside of our cells. Because the more these ROSs or ROSs build up, right, the more damage to other molecules your cell would incur. So one way that we deal with these ROSs or reactive oxidation species is through the use of this molecule, glutathione. All right, or GSH. So glutathione really is nothing more than 3 amino acids strung together. There's glutamic acid over here, right? You can see here's the carboxyl group, the amine group, here's the central carbon of this amino acid, and then this is the chain, the R group, right? That makes this a glutamate. Right? Then you have a cysteine over here in the middle. So here's its amine group, its carboxyl group, here's the central carbon, right? And remember, part of the R group chain off the cysteine has this sulfhydryl group, sulfur. That's what's involved in the sulfur-sulfur disulfide bonds between cysteine groups on different parts of, let's say, a protein that's wrapped around, right? Disulfide bridges really help to stabilize the proper tertiary three-dimensional structure of proteins. But here it's just a cysteine by itself, strung to a glutamate. And then this last one is a glycine, right? Super simple. It has an amine group, the carboxyl group, and its R group is just a hydrogen, right? So together, though, this is interesting. These 3 amino acids, we call them glutathione, right? And what glutathione does is it provides a nice mechanism by which you can neutralize reactive oxidation species. So here is a big enzyme that handles this process of neutralizing the ROSs using the glutathione. And so this enzyme is called glutathione S transferase, right? Or GST. So what does the S refer to? It refers to the sulfur group of the cysteine. And what is this enzyme going to do? It's going to transfer that sulfur group. Right? So here is the GSH here. Here is this nasty reactive oxidation species here. Right? This could be one of many different structures. The point is you have some structure that has an oxygen as part of it that is pretty dangerous, right? Because if this bumped into another molecule, this oxygen might want to react with that other molecule, potentially changing the structure of that other molecule. So what this GST enzyme does is it binds to the glutathione, and it will also bind to the oxygen group here, and it transfers the sulfur group onto this carbon. You see, so it's basically broken this oxygen bond here, and the oxygen ends up kind of going over to the right because now you've linked the glutathione to it over here. Right? Then the GST enzyme proceeds to remove the glutathione and replace it with another hydroxyl group. So in this form, these oxygens are far less likely now to react with other molecules inside of the cell. Right? So take-home message, we have this important enzyme, GST, that takes care of managing glutathione molecules as a convenient way to neutralize the various types of reactive oxidation species that are generated inside of our cells that, if they were just left alone, if they built up, could cause a lot of damage to other biological molecules. Not surprisingly then, if you have mutations that result in the loss of this GST enzyme, then you've lost the ability to properly control the buildup of these harmful reactive oxidation species inside the cell, and you end up with more cellular damage taking place, just as a byproduct of the cell doing its normal metabolic processes, right? Because remember, the GST enzyme, it's an enzyme, it's a protein, it's coded for by the GST gene that codes for it. So if you randomly have a hit, a mutation in that gene that messes up the ability to have functional GST enzyme, right? Your cell is still building up these reactive oxidation species, but there's no way to neutralize them. So let's look here. So here's a histology slide where we're looking at some prostate tissue. And what they've done is they've stained this tissue with an antibody that stains the GST enzyme in brown. All right, so the more brown you see is the more active GST enzyme in these cells to kind of try to neutralize these oxidation damage, right? And what you see is the normal basal epithelial cells over here. You can see the brown, right? And there's a lot of brown in some of these cells here, but look at where it's up against this sort of hyperplastic neoplasia. Right, so in these cells here, this is prostatic intraepithelial neoplasia. This is not cancer yet. This is still early on, right? But look at these cells that are becoming hyperplastic and beginning to lose control. You see low levels of the GST, right? So if this happens early on, then what's happening is these cells that are transforming are becoming more and more conducive to having more and more damage taking place inside of them. Right? And that damage can also, of course, be damage to the DNA sequences, right? As you have depurination, deamination, that's facilitated by all these reactive oxidation species building up. Right. Let's think about xenobiotic damage, right? Meaning stuff that we, eat, and then so it's introduced into our body as part of our normal diet. And then the exposure of our cells to the various types of compounds, chemicals that are part of the food that we're eating. That can be another way of thinking about, stuff that's coming actually from our cells processing things within them. Right? Our daily food consumption contains somewhere between 5,000 to 10,000, different types of natural compounds. Right. It's kind of interesting to think about because I think we usually don't think about like our typical diet, breakfast, lunch, and dinner in terms of like how many different compounds are we actually putting into our body and exposing our body's tissues to. So roughly, let's say 5,000 to 10,000 different types of natural compounds. Of those, we have about 2,000 milligrams or about 2 grams of burnt material. That just comes from the cooked food that we eat. We also have something like 1,500 milligrams or about 1.5 grams of naturally occurring toxins in the form of pesticides, plant products. Remember, plants that grow in the wild in Mother Nature, they've evolved to make all kinds of toxic compounds, right? Toxic compounds that are meant to discourage things like insects from eating them. Oftentimes, those compounds are poisonous to us too. That's why certain types of plants are actually very poisonous to us. An example I was just talking to my kids about was things like oleander, right? We have lots of oleander bushes out here in Hawaii, and they have really pretty flowers. Right? But oleander is actually very poisonous to people. Every year, you hear about cases of little kids, toddlers who end up getting poisoned because they're playing in their backyard and they're just eating stuff like the pretty flowers that come off of the oleander bushes, right? So again, right, those are natural compounds made by the plant to be poisons so that animals don't eat them, right? Including us. Other types of plant pesticides are poisonous to us, but we actually eat them on purpose and we enjoy them. Right? For example, caffeine that is made by plants, in particular things like coffee plants, right? What is caffeine? Do you ever think about that? Why does the plant make caffeine? Caffeine is a natural pesticide, right? Little bugs that come and try to drink the sap of these, like the coffee bean, right? They get a dose of caffeine, and caffeine ends up messing up neurons, right? Proper neurological signaling. So a dose of caffeine that's enough to kill a small little insect, is not going to kill us, but we can take a large dose of caffeine and we like the buzz, right? What is the buzz? Well, guess what? We're being poisoned by the caffeine, right? It's having an effect on our nerves and our neurons, but we like it though, right? I need my caffeine buzz in order to function. So my whole point is, before I go off like too far off on a tangent here, right, is that the food that we normally eat, right, a large component of it are kind of bad things already, right, that our cells are going to be exposed to as our cells process these types of things as part of their metabolism. What component of the food that we eat these days is actually synthetic, artificial poisons, such as artificial pesticides? You might think like, yeah, these days, right, so much of it is artificial stuff, right? You'd be surprised. Right, the average daily exposure to synthetic pesticides in food is only about 0.1 milligrams, right? So compared to the natural types of doses of compounds that could potentially damage our tissues, right, the synthetic stuff is actually quite small. So let's think about human development and human history, right, as part of our evolution. So humans started cooking food about 400,000 years ago. So less than 500,000 years ago is when, hominids developed, fire and the ability to cook foods. And, and I think all of us would agree that the ability to cook foods has made for much healthier foods, in the sense that you're killing, all kinds of, pathogens and things like that in the food, right? So food that might have been a little spoiled, if you cook it, right, you're not going to get sick, compared to food that was just eaten raw, that was not stored properly, right? But think about this. That means the introduction of burnt material as part of our diets, this two grams or so of burnt material that comes from cooking our food, that really only came along pretty recently in our evolutionary history, right? So 400,000 years compared to a few hundred millions of years of vertebrate evolution is very short. That means even though we're benefiting by cooking foods to be able to kill off germs and things like that, right, the big dose of burnt materials that we're taking in now, what impact does it have on our cancer burden, right? Is this actually an example of the way that we are, evolving our behaviors being too fast for evolution to really keep up? We've talked about BRCA1 as a protein that's very important for detecting things like double-stranded DNA breaks, right? I think several of you have chosen summary articles that talk about BRCA1. So BRCA1 is a very important tumor suppressor gene. So like many hereditary or familial, risks for developing cancer that's passed on in families, right? We said that certain tumor suppressor genes, you can inherit certain alleles that predispose you to developing certain types of cancer, like retinoblastoma was the first one that we looked at. So certain alleles of BRCA1 and 2 passed along through the germline, right, are definitely highly associated with increased risk of developing cancers. So for example, BRCA1, 2 germline mutations that are passed on from parent to child are responsible for over 50% of breast cancer cases that take place if you're talking about familial breast cancers, right? Breast cancers that run in the family can be attributed to having this particular allele of BRCA1 and 2. Ovarian cancer, it's even more pronounced, right? Over 70% of familial ovarian cancer cases can be linked to inheriting a particular BRCA1 or 2 mutation. Right? Sporadic mutations of BRCA1, meaning if you're born with wild type BRCA1, both copies, the one you got from mom and dad, right? If you get a sporadic bad hit in BRCA1, that accounts for over 20% of breast and ovarian cases that don't have like a familial hereditary history. So BRCA1 is very important for the detection of breaks in the double-stranded DNA that happen when you have certain types of chemical events, and let's say the DNA gets altered and broken, right? Things like, for example, X-ray radiation, right? They can actually break your DNA, your chromosome, and it's up to your cell to have BRCA1 as well as other proteins that can detect the double-stranded break and signal for P53 to go ahead and halt the cell processes so that other enzymes have a chance to try to repair the break. Right? And if the break is not repairable or there are too many breaks, then P53, rather than just halting the cell cycle based upon what BRCA1 is telling it, P53 might take the drastic step of pushing the nuclear button, right, and causing apoptosis to happen. But let's look at BRCA1 here. So this is BRCA1 in a cell culture experiment done in the lab, right, where they're staining BRCA1 in these cells. There's 2 cells here, and they're looking in the nucleus, and they're staining the BRCA1 with this green fluorescent marker. Right? So if the cells are just minding their own business, they produce BRCA1, and constantly there are incidents where maybe the DNA might break and BRCA1 goes there and helps to start to try to get some repair enzymes functioning. If you treat the cells, right, with a compound such as hydroxyurea, so hydroxyurea actually interferes with normal DNA replication and the processes of DNA maintenance. Right? What you'll see is suddenly the BRCA1 proteins begin to congregate in different places in the nucleus, where these double-stranded breaks are really becoming a big problem. Right? Likewise, if you look at, you say, well, what are these places where the BRCA1 is working, right? You can look at the presence of another protein, PCNA, right, which is proliferating cell nuclear antigen. So this is a protein that actually goes to DNA and recruits other proteins to help work on the DNA. And you can see here, sure enough, in the cells that are just sort of left alone, the PCNA is kind of spread out among the chromosomes in there. But if you go ahead and treat with the hydroxyurea, right, you can, it's treated with the hydroxyurea, so it actually is going to these different areas. Sorry, I didn't realize they don't have a minus hydroxyurea here. So the PCNA is also collecting in these particular regions. And if you overlap that with where the BRCA1 is, right, you can see the BRCA1 is really being drawn to the DNA by the PCNA, and it's trying to repair these defects in the chromosomes. So if you lose BRCA function, what happens? Right? You would end up with a lot of double-stranded breaks that aren't repaired properly, and importantly, you're not going to be properly signaling for P53 to put a halt to the cell cycle. So that these broken chromosomes then, right? Remember, if you break a chromosome in the middle, the ends have telomeres, right? But the part that breaks in the middle, you're not going to have telomeres there. So what would happen? If you have numerous chromosomes breaking, then they might stick together. And that's what you see here. You can really tell you have several chromosomes all joining up together improperly due to this loss of BRCA1 function. All right. So those are just things happening inside of our cells, either the normal metabolic processes of the cells, or our cells breaking down different types of normal components in our diet, right? So these are all things happening within our cells. What about exogenous mutagenic factors, things that are coming in from the outside? Here in Hawaii, we are very mindful of the sun, and we all love going to the beach and hanging out. We spend a lot more time out in the sun than certainly compared to some places on the mainland. So as a result, we're very aware of UV protection, right? So ultraviolet light is high energy wavelengths contained in sunlight that are constantly raining down on us as we're walking around outside. Right? And this UV light, that there's the longer UVA wavelengths and the shorter UVB wavelengths that can penetrate your skin. All right, so the UVA wavelengths, the longer ones, are usually associated, I think, with sort of the aging of skin, the type of damage that happens that causes, wrinkling of skin, things like that, right? Your skin getting more tough looking. The UVB is the one that penetrates deeper and actually can cause things like sunburn, right? And that's the one that's very dangerous because that's the one that has the real potential to damage your genome. Right? So when UV radiation penetrates our skin, right, and shines down through our corneal layer, this is our dead skin cell layer, into our epidermis, down to where our keratinocytes and our melanocytes are. Right? These radiation, the energy penetrates our cell's nucleus, and what happens is it causes cross-linking between adjacent pyrimidines. And so here you have, let's say, a strand of DNA, right? This is the phosphodiester bonds holding together these deoxyribosugars. And then you just have two examples here of two bases, right? So these two bases sticking off here. Right? These are purines. Oh, no, sorry, pyrimidines, right? These are pyrimidines, right? Like T's and C's, right? So let's imagine that we have over here, right? These two T's. What happens is that the radiation, it's energy, right? So it causes all the atoms to vibrate, right? Because that's what energy does, right? Energy causes the vibration of atoms. And as a result, it disrupts the normal bonds between these atoms temporarily, right? And hopefully the bonds will just reform back into their normal shape. But if there's enough energy, if you have these two pyrimidines next to each other, what can happen is you can actually shake free the carbon-carbon bonds that are taking place here and actually cause these carbons to improperly form bonds with the carbons of the adjacent pyrimidine, causing a cross-linking, right? So now this thing has become sort of a double base that's stuck here. Now you can imagine this is really bad, right? Because if you have enzymes that are riding along the DNA, for example, if this is in the coding region of a gene, right? Let's say of RNA polymerase 2 trying to read this, it gets here, it can't read through this, right? Because now this is like this weird double nitrogenous base there, right? So that's really bad. It needs to be taken care of. So how do our cells protect against UV exposure? Well, we produce melanin, right? So melanin is produced by melanocytes. And so there are different forms of melanin, but the one that we usually think about with our tanning is eumelanin. So here is a cross-section of our dermal layer. And what you have is mixed in here, these are mainly keratinocytes, but every so often you have melanocytes. I think this is meant to be a melanocyte over here. And the melanocytes, they detect that there is DNA damage taking place in the genomes. How do they detect it? The keratinocytes, right, are getting hit by UV radiation. And so you have certain enzymes inside of the cell that can detect that you're getting these pyrimidine crosslinks taking place, right? So certain enzymes will be the right shape to detect that. That will then trigger the cell to release certain types of signals that cause the melanocytes to go ahead and start producing eumelanin. And the eumelanin is actually released by the melanocytes and taken up by the keratinocytes. And the keratinocytes, it's actually kind of funny, right? They will actually carry or build up this melanin that they're taking in on the surface of the keratinocyte that is facing towards the sun. It's almost like they're putting on these little eumelanin hats on them, right? And so as more and more keratinocytes take up the melanin, right, we get our nice tan. So here's the eumelanin molecule. And what a crazy looking molecule, right? So this is what protects us against further UV damage. The way it works is if the energy from the UV radiation hits this molecule, the energy is basically absorbed by this molecule because of all of these double bonds through all of these rings. And so the energy is essentially absorbed and neutralized by this eumelanin. And I think eventually it just causes like an electron to like pop out or something like that to be discarded, right? So that way the UV radiation doesn't actually get to your DNA and cause these pyrimidine crosslinks from happening. So it's interesting to think about then, right? That means the fact that you are tanning is already a result of your genome being damaged in your keratinocytes. And that's when the keratinocytes then call for help. And the melanin and the melanocytes start making more melanin, and the keratinocytes take up the melanin to try to protect it from even more, right? So it's kind of weird if you think about it, right? So a nice even bronze tan really represents what? How extensive the genomic damage is throughout the surface of your skin. So in strong sunlight, it's estimated that you can actually accrue over 100,000 pyrimidine dimers in your genome per hour as you're lounging there on the beach, right? And if that's not enough, we're not talking about your whole body. We're talking about per cell, right, that's exposed to the strong sunlight. Right? So if tanning is our natural response to UV exposure, and so again, I'm challenging you to think about this. You know, we tend to think of a nice tan as being a sign of being healthy, right? I mean, it is a sign of being healthy in the sense that your body is responding to widespread genomic damage the way it's supposed to, right? And also because, you know, being part of being fit and healthy is, you know, being able to be out in the sun and being able to produce your vitamins from the sunlight, things like that. But if tanning is our natural response to UV exposure, you know, then what is sunburn? Right? Look at this guy. Right? So rather than staying inside and attending biology of cancer class on Zoom, he said, "I forget this. I can always watch it later on YouTube. I'm going to go hit the beach." Right? What a loser. Okay, so anyhow, don't be like this guy, right? So what is sunburn then? I think when I was a little kid, I actually took it quite literally. I took it to mean like, oh, the sun is so strong, it's like burning your skin, right? But what is it? Sunburn is when the keratinocytes realize that they have way more DNA damage than they can repair. So rather than just worrying about getting more melanin, right, they cannot fix their extensive DNA damage. P53 pushes the self-destruct button, and they undergo apoptosis to prevent cancer. So that's why, I mean, you all know this, right? That sunburned skin is skin that's on its way to dying, right? That's why it ends up, if you have really bad sunburn, right, it ends up peeling. Right? Billions of your cells are choosing to die so that you can continue living, right? I mean, you look at this guy, and all of us have had sunburn before. How many billions of cells does that represent that have gotten too much genomic DNA damage that cannot be repaired? So they choose apoptosis rather than trying to keep surviving, right? So that you, as the whole organism, the whole body, can continue living free of cancer, right? Or at least free of the consequences of extensive DNA mutations. But if billions of your cells are choosing to die and doing the right thing, does every single affected cell actually go through with it? Right? Do all of them manage to undergo apoptosis properly? What if in reality there's a handful, right, that don't undergo apoptosis, right, and those cells start transforming? Maybe because as part of the random DNA damage that they've gotten from the strong UV sunlight exposure, maybe certain genes that are part of the apoptosis pathway have been mutated, right? So that apoptosis is not happening as it should. Okay. So when we're talking about things like, exposure to things from the outside, I mentioned that oftentimes we think of food products, burnt products, you can think of it both as being something that's chemically coming from inside your cells because you're eating food and it's processing it. But of course, you can also think about it as external exposures, right? Things like contained in charred meats, for example. Hey, I always love a nice steak on the grill, right? And we all sort of love the taste of char grilled stuff. I don't know why exactly. I think probably because when you burn certain types of foods, whether it's meats or vegetables, whatever, there are certain types of aromatic compounds that are released as the chemical nature of the food is altered by heat, right? And so we've kind of learned to develop a real liking for that. Right? So but when you're grilling foods and you're releasing all kinds of stuff in smoke and in the burnt parts, you are often talking about aromatic compounds, right? Aromatic meaning made up of ringed structures that are created from the heat, disrupting the normal chemical structure of these food molecules. So things like benzoapyrenes. So if you look here, this is a structure of a benzoapyrene that can be created in grilled foods that are kind of burnt. Right? And these can form what we call chemical adducts, so ADDUCT, with the molecules inside of your cells. So in this case, look, this benzoapyrene ended up forming an adduct. It got linked onto this particular nucleotide, this guanine here. Right? And as a result, this guanine nucleotide now has this big, bulky thing sticking off of it. Right? Look at the structure of the DNA over here. Right? So here's the nucleotide, and here's the big BPE sticking off of it. It's very clear, right, that this disrupts the normal alpha helical shape of your DNA. And if this adduct cannot be resolved, meaning removed, right, you've got a big problem here, right, because this information that's contained in the DNA here is compromised. So one way that our tissues have evolved to protect against things like these exogenous threats, right? Whether you're talking about smoke that you're breathing in, right, or UV exposure. One thing that you can think about is, well, yeah, I guess if certain cells are positioned deeper in our tissues, then that affords a particular level of more protection, right? Now, not all of our cells can be buried deep away from exposure to things. Think about all the differentiated cells that line our skin, our GI tract, and our mucosal layers, things like that. Those fully differentiated cells are right at the front line of having to take the brunt of a lot of these types of mutagenic factors, right? But what about the stem cells? The stem cells can be buried deeper in our tissues. And if you think about this, right, we've learned that the differentiated cells, we're actually okay with them getting damaged. Why? We accept the fact that differentiated cells accrue a lot of damage because they are exposed to carcinogens, mutagens as part of their normal function. And we're okay with that because many of those fully differentiated cells, especially epithelial cells, right, the focus of what we sort of talk about in this class, right, we just lose them within a short amount of time anyhow, right? So even if they're building up lots of mutations, whatever, right? A cell that's lining your small intestines, even though it gets some hits from chemicals in your foods and it has random mutations, right? It's starting to transform, whatever, right? 5-6 days later, your body loses it anyhow. But your stem cells, you need to protect because your stem cells are in charge of repopulating all of those differentiated cells. So look at this figure here. Stem cells are shown here in this blue compartment, which is deeper, right, away from the surface of where your differentiated cells are and where those cells are being exposed to mutagens. Remember that these stem cells, their job, right, is to just every so often divide as part of repopulating all those differentiated cells that are lost. So here, let's imagine this one blue stem cell, it divides, right, and it makes 2 daughter cells. One daughter cell becomes this green cell that starts becoming this rapidly dividing set of transit amplifying cells, right? And along the way, they differentiate. They begin to carry out specific gene programs that lead them to become many, many highly differentiated cells from this one daughter cell here. The other daughter cell that resulted from the stem cell dividing is this one, right? So this one maintains its stem cell properties, right? So that's when we say that a stem cell persists. What we're really saying is every time it divides, one of the daughter cells carries out the function of becoming differentiated cells. The other daughter cell, right, holds onto its stem cell-like properties so that when the time comes, it can divide again, right? And et cetera, and et cetera, and et cetera. Right? So part of the protection is being buried deep in the tissue or deeper in the tissue. The other part of it is it's really not proliferating that rapidly, right? It's very occasional because every division gives rise to many, many fully differentiated cells. Right. So stem cells are protected from mutations by their physical location away from mutagens, their relative infrequency of division. Right? After all, every time a stem cell divides, it's going to get more mutations just through mistakes in the DNA replication process. Right? Thirdly, stem cells have certain properties to them that have to do with being primed to undergo apoptosis. So in other words, if a stem cell begins to detect that there's DNA damage to it, right, you know, other cells might try to repair the damage, right? But stem cell is more likely just to undergo apoptosis because it doesn't want to risk it, right? If there's damage, what if it can't repair the damage properly? It's more willing just to undergo apoptosis and take itself out of the picture. And stem cells express different sets of proteins that help protect it. For example, different types of transmembrane pumps. So there are different types of proteins that serve as pumps that pump different contents of the cell out. So if small molecules get into the cell, these proteins pump them back out of the cell. So these might be certain types of toxins that get in. So stem cells are way better at pumping them out. Right, these membrane pumps. And lastly, I just want to throw this out there. This is the last thing for today. There's also this interesting kind of concept that not everybody believes in, but it's called asymmetrical allotment of DNA to the daughter cells. Remember, every time DNA gets copied, remember how DNA, the double-stranded helix, when it gets replicated, it is a semi-conservative process, right? The two original strands each become a template, right, to make a new strand, right? So that the two molecules that form actually still have the original strands as part of them. Right? After that second round of replication, right, you actually end up with some strands that are new compared to what you started with, right? But yet the two original strands can still be part of subsequent divisions. So there is this kind of idea that maybe, right, because the mistakes that happen during DNA replication are reflected in the copy that's made, not the original strand, right? So there's a thought that maybe stem cells preferentially hang on to the original strands of DNA, and the daughter cell that becomes the one that goes on to form the transit amplifying cells, it just kind of gets the copies, right? Whereas the daughter cell that still has the original older strands of DNA is the one that tends to stick around as the stem cell, right? And that has not been proven because I think it's very hard to try to follow that, right, in real life. But it's an interesting way of thinking about it, right? So it's kind of controversial whether or not that's true or not. Okay. All right, let's go ahead and stop there today.